Thank you for joining us today as we kick off National Tick Awareness Month 2020. This year's theme is Tick Season Can Be Every Season. My name is Nicole, and on behalf of the Canadian Veterinary Medical Association and Merck Animal Health, I would like to thank you for joining us today as Dr. Robbin Lindsay and Dr. Clow discuss tick seasonality, provide an update on tick expansion in Canada, review changes in tick-borne disease risks over time, and explore the role of the veterinary community in One Health. In support of National Tick Awareness Month 2020, all clinics across Canada will be receiving or have already received a poster to display in your clinic and a letter which provides information regarding the tools available to promote tick awareness. If you would like more information about National Tick Awareness Month campaign materials for 2020, please go to the resources icon at the bottom of the screen, which contains the social media toolkit and the campaign poster. Additional resources can be found on the CVMA website. As well, we invite you to visit the updated www.ticktalkcanada.com website, which provides valuable tick education materials and videos for your pet owners. Before we begin, I would like to read a few words from Dr. Melanie Hicks, the president of the CVMA. Dr. Hicks writes, "Thank you all for joining us for this very important webinar. The shifting geographic distribution of black-legged ticks through regions of Canada has been a real game changer for veterinary teams and our clients. This rapid expansion creates a growing need for us to educate our clients and update our parasite control protocols regularly. We've seen that in some parts of Canada, black-legged ticks can be active year-round. So it is important that we take the time to educate our clients on how best to protect their pets and themselves from parasites such as ticks to help mitigate the threat of zoonoses such as Lyme disease. Thank you again for participating in today's webinar, and thank you to Merck Animal Health for helping to continue to support the National Tick Awareness Month for its 5th year. Today we are fortunate to have two experts in their field. Dr. Robbin Lindsay received his bachelor's degree from the University of Winnipeg, a master's degree from the University of Manitoba, and his PhD from the University of Guelph. He currently is employed with the Public Health Agency of Canada at the National Microbiology Laboratory in the Zoonotic Disease and Special Pathogens section. The focus of his work is laboratory and field-based surveillance for various zoonotic disease agents, including hantaviruses, West Nile virus, and other vector-borne infections like Lyme disease. Dr. Clow is a graduate of the Ontario Veterinary College and currently holds the position of Assistant Professor in the Department of Population Medicine at the college. Her research focuses on the ecology and epidemiology of zoonotic and vector-borne diseases. She is passionate about One Health and eco-health and strives to make a positive difference in the health of animals, humans, and the environment. And now, I'll pass it over to Dr. Lindsay to get started with today's webinar.
Excellent. Thank you very much. My name is Robbin Lindsay, and I'm going to talk about the range expansion of ticks and some of the dynamics in the change in pathogens in those ticks. And then I'm going to hand over to Katie Clow from the University of Guelph, and she's going to talk more on the One Health aspects of the tick work, and she's also going to provide some background on some of the excellent work she's doing. Before we can discuss the changes in the range distribution of, or the range of black-legged ticks, I think it's important that we do understand a little bit more about the biology so you can have a better sense of what's driving a change in tick populations. There are relatively few ticks present in Canada. We only have about 40 species of the close to 900 species present. Many of these are quite common that we see many of the Ixodes and Dermacenter out there, but they are at the northern limit of their distribution. So all of them to some degree have to overcome the period of dormancy that is caused by winter. So that's an additional challenge that ticks in Canada have to face. With respect to the types of ticks we see, although we have 40 different types present in Canada, there are really only about 7 or 8 different types that you're likely encounter, and they are illustrated here with the black-legged tick and its cousin, the western black-legged tick. It looks identical to this. The groundhog tick. There's 3 types of Dermacenter. The 2 that are most common are the American dog tick, pictured here, and it looks identical to the Rocky Mountain wood tick. We are seeing more Lone Star ticks, and we also see brown dog ticks and rabbit ticks. So of that very diverse 40 species, there are really only about 6 or 8 that we're interested in understanding the distribution. From a human standpoint, it's even fewer because there's only about 6 species that bite people. So we're interested in the ones that are going to potentially bite people and transmit pathogens to them. Now, ticks, of course, are obligate blood-sucking ectoparasites. They require blood in order to molt to the next stage. So a larvae takes a blood meal, becomes a nymph. Nymph takes a blood meal, becomes an adult male or female. And the female requires blood in order to lay eggs. Male is there to inseminate the female so she'll lay viable eggs. Now, how do ticks find a host? Again, the blood is essential to their development. So they really have 2 basic strategies. They can be nest associates, so they're present in the nest of the host animal they feed on. This makes host acquisition much easier, provided the host doesn't abandon the nest. A number of the species that we're interested in also participate in this behavior known as questing, where they seek a host in the environment by climbing onto vegetation, as is illustrated here by black-legged tick here and winter ticks on a blade of grass here. And this represents an additional challenge to these ticks. If they're not in the right environment, they won't have the appropriate questing substrate to get to the host that they're interested in. And of course, this is a prone period for them too. They're out in the environment. And again, it's at this point that they're prone to desiccation. If the tick is lucky enough to encounter host, then it uses the business end of the mouth parts here to penetrate the skin. And they usually do that by introducing an anesthetic because they're going to feed for long periods of time. Typically, with a black-legged tick, for example, it's larvae and nymphs take 3 to 5 days to engorge, and then adults take anywhere from 5 to 7 days, and the other species take even longer to feed as adults. When they drop off, then they have other developmental stages that are influenced by temperature. They go from a fed larvae to an unfed nymph, and they go from an unfed or fed nymph to an adult is temperature dependent. Females depositing or maturing eggs, depositing those eggs, and then hatching is also dependent on temperature. So all of these life history events usually extend the life history of the tick. Most species require at least 1 year developed from to complete one generation. Most of the ticks, though, in Canada are at least a 2-year, if not a longer, life history. In terms of the seasonality, that's also quite variable. The peak season for activity varies between the species. You think of American dog ticks and Rocky Mountain wood ticks, they're really a spring or summer species. Compare that to the black-legged tick here, where the adults become active in the late fall. There's also differences in the life stages. It's illustrated here by this diagram that shows that the larvae of the black-legged tick occurs in the early fall. The nymphs are present during the summer months, and then again the adults present in the fall. If they have to overwinter, they would become again active in the spring. And what regulates those activity periods is sometimes it's under the influence of diapause, but certainly environmental conditions determine whether they're capable of being active as well. Most species have a minimum temperature threshold in around 4 degrees. So when the temperature drops below 4 degrees, they're no longer capable of extended activity. Certainly snow cover on the ground as well. If they cover up those questing substrates, then they can't be active. So across the country, we can see a tick like black-legged tick. It's very unlikely to be active in January in Manitoba, but in Nova Scotia, where they can get periods of that snow is reduced or temperatures are above 5 degrees in January, they can be active. So it varies on a regional basis. And you can see that peak periods of activity are important in terms of transmission dynamics here, because the nymphs of the black-legged tick and the adults are the stage that transmits, and these are the periods when we're at greatest risk. In terms of the overall distribution, you can see that some ticks in Canada are found from coast to coast. Others are found just during or only found in the coastal regions. Some have a primary western distribution and some have just an eastern distribution. And again, part of that is a function of their host preference. Dermacenter albopictus, or the winter tick, is found on moose. So the moose are widely distributed, so the tick is widely distributed. Likewise, Ixodes uriae is a seabird tick, so it's only going to be found on seabirds. So again, whether their range can expand is at least in part a function of how abundant their hosts are for them to potentially realize that expansion. And the black-legged tick shows you a good example of the diversity of hosts that can be involved in feeding. Here, the larvae of the black-legged tick feed on small mammals with a preference for white-footed mice or shrews and voles and the like. They can also feed on birds, so that's a potential dispersion mechanism. The nymphs of that species will also feed on those same small mammal species, and again, that provides an opportunity for amplification of pathogens within an enzootic cycle of transmission. If both stages feed on the same host type, they can transmit during one stage and acquire it back from the host in the next. And what's interesting too is in black-legged ticks, the adult stages never feed on these small mammal hosts or birds. They're found on medium-sized mammals and have a preference for deer. So if a tick population is going to expand into new areas, it has to support the small mammals and the medium-sized mammals in order for the population to become established in a given area. Of course, habitat is very important as well. Ticks across the board have some differences in their habitat preferences. You think about American dog ticks or Rocky Mountain wood ticks, they're really a prairie species or a montane species, whereas black-legged ticks do much better in forested habitats here. They really require or do much better in areas where there's a full forest canopy. Any tick species can incur into a less than optimal habitat and be present there, but typically they don't do as well if they're not in the habitat of the preferred or required habitat. And again, what we see with many species, including the black-legged tick, is the deciduous trees dropping those leaf litter creates a moist microhabitat where lots of the off-host behaviors and development occurs, from females laying eggs in the leaf litter to those eggs hatching into larvae and all the unfed stages rehydrating within this substrate. So having appropriate habitat creates this area for them to be able to extend their survivorship. And of course, climate in general is an important parameter for and has an influence on limiting the distribution of some species. We've been able to show that you can think of ticks as like corn. They require a certain number of degree days above a threshold for development. If an area doesn't provide that, then that's going to be limiting towards in terms of their potential distribution. And again, my colleague, Dr. Nicholas Ogden, did some neat work where he looked at the predictive, based on the anticipated change or climatic warming patterns. He estimated that the climatic suitability, as I'm just cursoring through here, will increase over time, in much of eastern Canada. So the habitat suitability, the climatic suitability is going to improve, but it comes almost a bit of a moot point here that it's going to get as far north as we see here, because 90% of the Canadian population lives within 100 kilometers of the US border. So it doesn't take much of an incursion and establishment into Canada to put a lot of people and companion animals at risk of exposure to these particular ticks. And of course, these ticks need, because I say they don't really disperse on their own, but ticks can fly. If they get on a bird, they can fly. They can also move, tens of kilometers on animals such as wolves or coyotes. Deer migrating or moving during the rut can move them, 20, 30 kilometers at a time as well. We do a lot to move our ticks around through moving of infested companion animals and livestock. So these are the factors we need to consider when we're looking at the change in distribution. And again, one of the best examples of dramatic range expansion is illustrated by the black-legged tick. Now, this tick was known from the Long Point Peninsula. It's a spit that sticks into Lake Erie, south of Simcoe. This population of ticks was known and studied during the 70s. It was probably there for decades prior to that, but right around the time that Lyme disease was emerging, and subsequently, we wanted to look at this population because it was the only location where the ticks were established. And I was lucky enough to be involved in a research project where we looked at the factors that limited the distribution of black-legged ticks in the environment of that location. And what we looked at was, the environmental conditions, the habitat types, and then we looked at the abundance of ticks. They're small mammals, deer in this environment. Try to get a sense of, and what we were observing was ticks are present in Long Point, but there were certainly suitable or similar habitats along the north shore of Lake Erie where these ticks didn't appear to be. And Dr. Ian Barker did some surveillance through much of southern Ontario and really came to the conclusion that for some reason these black-legged ticks were isolated or found primarily on Long Point and not other locations. And what we saw was, and again, this map, hopefully you can see it better than it's showing up on my screen here, but through the sort of early 90s, we saw focal populations of these ticks establishing in areas similar to Long Point. So John Scott and our group detected ticks at Rondeau Provincial Park and Point Pelee, again, sand spits that stuck out into Lake Erie, similar habitats. But it was around the 2000s here, early 2000s, that you really started to see a tremendous change in the distribution of ticks, where they went from relatively small numbers of populations to increased and large numbers of them right through much of eastern Canada. And what we typically saw was you could have isolated populations of ticks that would establish in a given area, such as Lunenburg. That was a population detected in 2002. And the ticks subsequently spread at a local level in those habitats. And then subsequent populations were detected in those locations as well. So we see quite a difference in terms of the way the ticks were establishing and the speed with which they were establishing through the early 2000s. And you can see this is the distribution of the risk areas approximately now. And this is phenomenal change in the range of these ticks, from having only one population in 2002 in virtually every jurisdiction in Nova Scotia or counties there, now with established populations. We found ticks in New Brunswick in 2008, and now they're present in much of the south shore of that jurisdiction. Tremendous population establishment in southwestern Quebec and eastern Ontario. And again, this was likely occurred just from sort of 2000, mid-2000s onward. Southern Ontario, you see much less in terms of more focal populations, in probably driven by the types of habitat and host availability in those locations. And even in some urban centers, you look at the Rouge Valley in Toronto, it's a beautiful riparian habitat that goes through the city. Again, it's a sort of a specific habitat within a large metropolitan area that is capable of supporting ticks. And in Manitoba and northwestern Ontario, we see the same sort of pattern, where we had an incursion of ticks in one location and then subsequent expansion. And again, we think in this particular instance, maybe spillover from endemic areas for black-legged ticks in Minnesota, moving up to the border and then spilling over, and then transport in a westerly direction, again, along in habitats that suit the tick. We think that these ticks will get into Saskatchewan and perhaps into Alberta, but the habitat available to them in those locations is reduced, so we're probably not going to see the same level of activity. And Western black-legged ticks are quite a bit different. They are already fairly widely distributed in British Columbia, and we don't see the same traumatic range change that we see with black-legged ticks or Ixodes scapularis. Okay, so range expansion is not just occurring with black-legged ticks. We see a similar pattern with American dog ticks, and this is some data from my colleagues in Nova Scotia. They show from the 40s here, this tick, based on the surveillance that they've done, has expanded its range in western portions of the province through through the early and late 90s, all the way into the 2000s. So range expansion is occurring with other species, not just black-legged ticks. And this pattern of range change for American dog ticks is likely duplicated in Manitoba and Saskatchewan. So that we're seeing a similar pattern of range expansion. And why do we care? We care because ticks cause a number of conditions here, but primarily they're the transmitters of pathogens of human and animal health importance. Again, in Canada, we see a number of infections that are transmitted by ticks, and the most important ones here are primarily transmitted by Ixodes scapularis and Ixodes pacificus, with a bit of a input from other species. But these are the primary ones. They also transmit Anaplasma and Babesia. And there's an interesting lineage of Powassan virus called deer tick virus that's being observed in black-legged ticks. And we also have the classical Powassan virus transmission cycle involving Ixodes cooki or groundhog ticks and Ixodes marxi. The other tick-borne pathogens there seem to be much rarer and are transmitted primarily by either Rocky Mountain wood ticks or American dog ticks, with an occasional one soft tick transmitted infection known as relapsing fever. So overall, what we're seeing here is the greatest change is a function of, in terms of human health risk, is that of Lyme disease. And really it's driven by two things: the transmission of, the acquisition of, or prevalence of infection in these ticks being at a fairly high rate, and the tremendous range expansion. So this pathogen is important because it's in a high rate of prevalence in the ticks, and these ticks are changing in distribution, but they're not the only ones that are changing in transmission dynamics. Anaplasma phagocytophilum is found in black-legged tick populations in every province. Ticks are infected at about 1 to 4%. And we have seen case reports in animals, we've seen them in humans, and the range seems to be expanding. Babesia microti is also a pathogen that we see expanding in certain tick populations, mostly in Manitoba. But Powassan virus is found in populations in Nova Scotia, in New Brunswick, in Quebec, Ontario, and Manitoba. So it is one that we are interested in. And the top three here, we're so interested in them, we're trying to include them in the nationally notifiable disease list so we can get a better understanding of how these pathogens are changing over time and what that impact is for human health. I think that's it for me. I'll just hand that over to Katie, and she can talk a little bit more about the importance in terms of animal health.
Great. Can you hear me okay? I see a nod from Robin. Awesome. Okay. Well, thank you everyone for inviting me to this national webinar. I'm going to build on a lot of what Dr. Lindsay has already said, and specifically talk about a national study that we have underway called the Canadian Peptic Survey. And so I'll just go over a little bit more about the rationale of why we're doing this survey, look at our approach, some very preliminary results and take-home messages, and then finish up by talking about One Health and how veterinarians can have a key role in a One Health approach, particularly with ticks and tick-borne diseases. So as Dr. Lindsay had already mentioned, we're seeing dramatic changes in our tick populations and ongoing range expansion. And this is evidenced particularly by the black-legged tick and the American dog tick. But there are also other, as we say, non-native tick species that we are monitoring for incursion and invasion into Canada. And the one which you've probably heard about quite a bit, and Dr. Lindsay did mention, is the Lone Star tick. It's in the media all the time because it can infamously cause a red meat allergy. This tick has undergone significant range expansion in the United States. Used to really predominantly be viewed as a South Central United States tick, but as you can see from the map on the left-hand side published by the CDC, this tick has expanded all the way up through the northeastern seaboards and can be found in several bordering states to Canada. And there are some work that's been done looking at predictive modeling in the context of climate change and how this tick may expand its range in that context. And you see all of the areas on the right-hand map of really dark red in southern Ontario and into some of our maritime provinces, where we know that the climate will become climatically suitable for these tick species. We do see Lone Star ticks introduced every year on migratory birds. And if we think about ecology of this tick species, we also know that we have lots of good hosts for this tick species. They love white-tailed deer and wild turkeys, which anyone in Ontario knows we have a ton of those, but is a pretty indiscriminate feeder and will happily find its way on other mammal species, including ourselves and our companion animals. And it's a really aggressive feeder and is a vector for several pathogens of human and animal health significance. So it's something that we have been keeping a close eye on and want to do continued monitoring for. Another tick species, which you may have heard of, which is also on our radar for invasion into Canada, is the Asian longhorn tick. And this tick has a very interesting story in that it was found for the first time in North America on a sheep farm in New Jersey in 2017. I think everybody in the tick world really hoped that this was just a one-off mistake and that the tick wouldn't overwinter. But subsequently in the spring and the following years, it's been found in almost a dozen other states. And really, we don't know how it got here. There are, I think, lots of theories, but there's evidence that it's been here longer than since 2017. As the name suggests, from Asian longhorn tick, it's native to Asia, and it's also invasive in Australia and New Zealand, but never before found in North America. This tick has a pretty interesting property in that a female does not need a male to reproduce. And this is different than any other tick species that we're dealing with, and thus can invade and spread quite quickly. And so on this map, on this slide, you see the red plus signs are places where this tick has been found. And then in between, overshadowed in the blacks and the grays, are areas where the climate and habitat is suitable to support this tick species. And so certainly in areas of southern Ontario and the maritime provinces, there are many areas where this tick could sort of set up shop. And because of this ability to reproduce without a male, it is quite an invasive species, and so something that we're particularly concerned about. Right now, we don't know a lot about the pathogen risk, particularly in North America, but as a pest, it is particularly deadly to livestock and can form high infestations and has been reported to have led to exsanguination and death of several cattle in the United States, and so something that is pretty scary and something that we want to keep an eye on. And then finally, as Dr. Lindsay had mentioned, we also know that the dermacenter species are changing their range in Canada. And recently talked about with a similar study than ours in the United States is if you look at the two maps on the left-hand side, you sort of see this clear demarcation in the States where we see the American dog tick in the east and the Rocky Mountain wood tick in the west. And surveillance done in this area where the Rocky Mountain wood tick was or is, all the ticks submitted from that region except one were actually American dog ticks. And so this is recent work that has come out of Dr. Susan Little's group and sort of showing that ongoing range expansion of the American dog tick into areas where the Rocky Mountain wood tick is. And we've seen some of this early spread by Sean DeGursov's and colleagues' team looking at the tick species in Alberta and Saskatchewan. And so something that we want to continue monitoring as well as seeing which dermacenter species are present in our western provinces. And so taking all of that into consideration has really been a rationale for our Canadian Pet Tick Survey. And so this was a national, or is a national study of ticks on companion animals, and we launched it last April, and it is continuing into the end of March, so for 1 year. So we're in our last month of submissions. We have 94 clinics across Canada that are in always budgetary constraints. We have generous funding by Pet Trust at the Ontario Veterinary College, And the study is divided into 3 key components. So we complete tick identification of all of the samples, pathogen testing, and then we're also collecting information on tick infestation patterns. And so there are a few study objectives that we wanted. And first is to establish a comprehensive current baseline of distributions on dogs and cats across space and time. And where the real richness in this study is the ability to get regionally specific data. And so not just sort of at the provincial level, but looking at, since we have large geographic areas, different regions across Canada to understand what patterns we're seeing. The next is to assess patterns of tick species infestations on dogs and cats, and I'll give you a bit more information about what we mean by that in a second. The third, to determine the pathogen risk from these ticks, and then really looking at applying a One Health approach so that we can work with and share our findings with human and environmental health colleagues and really create a holistic approach across ticks and tick-borne disease surveillance. So this is a basic map showing the location of our clinics. And so as you can see, we have clinics submitting from every province across Ontario. This was mostly a convenient sample, so sort of a first come, first serve enrollment. But we did certainly reserve certain numbers so that we could get representation from all provinces and across different areas and provinces. And so we have really strong representation from British Columbia, Ontario, Quebec, Nova Scotia, a little less throughout the West, but we still have representation from all of our provinces. And really, we're hoping to get good regional data from having this many clinics submit to us. And so how this study works is that we have prepaid, pre-addressed envelopes and vials that have gone out to all the clinics, and they're encouraged to submit all ticks for an entire year from companion animals. And these ticks are submitted in batches, which reduces our postal costs and allows more ticks to be sent in. And with each tick sample, it's accompanied by this tick submission sheet, which is giving us some basic data on date and location of tick acquisition, pet travel patterns, of course, pet demographic information, and then a little bit on preventative measures, so looking at if a tick check is done and if the animal's on tick prevention, and then also location of tick bite. And as I said, there's a similar study being conducted right now in the United States by Dr. Susan Little's group at Oklahoma State University. Some of their preliminary results are really strongly showing certain ticks are feeding on certain areas of pets' bodies. And so we want to collect data on this because, as you can see, if there's really strong relationships there, we can communicate that to owners on the most appropriate places to be doing tick checks, especially if you're limited in time or have a really hairy dog, sort of the biggest bang for your buck type approach. And so when our samples come in, the batches are processed and everyone receives a unique identification number. And then the first thing that happens is ticks are identified with a stereoscope using standard keys. Very early on in our study, we realized that we were getting a mix of dermacenter species from the West, and they can be a little bit challenging to identify underneath a microscope between Dermacenter variabilis and Dermacenter andersoni, especially for our lab because we really hadn't gotten very many Dermacenter andersoni in the past. And so we made a decision that we would, for all the species that come from the West, we would do PCR confirmation of the species just to make sure that to validate our stereoscopic identification. Ticks are pooled if they come from the same pet, are of the same species, life stage, and sex, to a maximum of 5 ticks per pool. And that's really just to help with the financial cost of testing ticks. And finally, all that DNA is extracted, and then we do pathogen testing. This chart just shows the key pathogens that we're looking for, and of course, they're specific to tick species. For Ixodes scapularis and Ixodes pacificus, we're looking at Borrelia burgdorferi and Anaplasma phagocytophilum. As Dr. Lindsay had mentioned, there are other pathogens that these ticks can carry. Because this is a pet-specific study funded through the Pet Trust Charities, we decided to focus specifically on pathogens that will affect companion animals. That being said, because we are extracting and keeping all of the DNA from all of these ticks, there may be the potential in the future, if we can access some additional funding, to look at other pathogens that are human specific to really make the most of the data and the ticks that we're collecting. But right now, this is our procedure. For our germicenter species, we're looking at Rickettsia rickettsii, which is the causative agent of Rocky Mountain spotted fever, as well as Cytozoon felis, which causes cytozoonosis in cats. And then in our Amblyomma americanum submissions, we're testing for 2 different types of Ehrlichia, and then again, Cytozoon felis. And as you can see, these are our protocols that we're following in the lab. And so now I'm going to jump into some preliminary results. I'd like to put a caveat that we've gotten a tremendous amount of data from this project, which is fabulous and exciting, but we have not had the ability to process all of it to date. And so you will see that as I'm presenting preliminary results, it'll say how many samples those results include and up to what date. And also there's some restrictions on any conclusions we can make, because some of our testing, particularly our pathogen testing, is still a very small sample size and doesn't have, we haven't dug down to the regionally specific level. So they're just very superficial results currently. So the first thing that is a big pattern that's come out of our study is about 90% of the ticks submitted are from dogs and 10% are from cats. Although this seems like, well, that's not very many on cats, there's sort of a preconceived notion, I think, within our profession that cats actively groom ticks off, and we really don't see ticks on cats. And really, this study is showing that that's not true. 10% of, over 2,500 submissions is still a lot of cats that are having ticks on them. And so it's just sort of a further evidence that depending on our lifestyle choices for our kitty cat friends, we need to pay attention to whether tick discussions are important for that pet. This is our species distribution so far. So this is the number of ticks submitted by species as of December 1st. And that includes over 4,100 ticks. And that's not 4,100 submissions. It's probably about 2,500 submissions for those numbers, because some pets have multiple ticks on them. But as you can see, we're about almost half of our submissions are Ixodes scapularis, and the other half are germicenter species. And then sort of the runners up, we also have some Ixodes pacificus, which are coming from British Columbia, a portion that are Ixodes cookii, so those are our groundhog ticks. And then up here we have a small portion that are all sorts of other species, which we'll look at right now. And so our other species cover a range of genuses. The highest number of sort of other species that we're seeing are brown dog ticks, which in some ways seems surprising because these are not ticks that we find in Canada. They're predominantly in tropical and temperate environments around the world, but really places that are not quite as cold as Canada. But this tick is unique in that it can happily survive indoors for its entire life cycle, and form massive infestations in households. And what we're seeing is that pets that have traveled are bringing these ticks in on them, as well as dogs that have been rescued from international areas, particularly in temperate and tropical regions. And the sort of concerning factor about this is that these ticks, as I said, can form infestations. And if they're introduced into a household really before the problem is noticed, by the time you see ticks crawling up your wall, it's a little bit too late. And at that point, massive pest control is needed to be brought in, and they're pretty nasty. They can carry a number of different pathogens. And so this is really just ongoing evidence to really support educational conversations with any rescue groups or people who are considering international rescue, that this is just another risk, and to use appropriate prevention strategies before that animal comes into Canada. We've seen only a few submissions of Lone Star ticks, which is reassuring. I think 2 of these 3 submissions all have travel history into the States. And so we're not seeing any patterns in this current submission cycle that is indicative that many of these ticks are being found within one region and of different life different life stages, that would make us think maybe something different is going on and there's a reproducing population. And so right now, these Lone Star tick findings are really consistent with adventitious or travel-associated tick findings. We've seen a few rabbit ticks as well. Whenever we see a tick that has a headpiece that looks like that, we get a bit concerned because, as I mentioned, the Asian longhorn tick is a Haemaphysalis species, but all of the Haemaphysalis ticks we've gotten so far have been consistent with rabbit ticks, which we find natively in Canada. Then down at the bottom, we've seen a bunch of other species, so some Gulf Coast ticks, a bunch of more wildlife-associated Ixodes species, and then one submission of soft ticks, which I screeched a little when I saw them underneath the microscope because they're pretty creepy looking. But and that was the spinous ear tick from British Columbia. And we can look at these results by province. And again, as I said, when we sort of hone these down into a more regional level and put them on a time scale, we're going to get some really good data out of that. But right now, this is a pretty superficial level. As you can see, our Dermacentra species are a large proportion of the samples we've gotten, particularly from British Columbia, Saskatchewan and Manitoba, and Nova Scotia. And then our Ixodes scapularis is dominating in Ontario and Quebec and New Brunswick. The other species are shown in green, and as you can see, particularly in New Brunswick and Quebec, there's quite a number of other species. The majority of these are Ixodes cookii or groundhog ticks, which are common in that area of Canada. If we look at some of our pathogen, PCR for species, we see some interesting findings. And again, as I said, this is only a small number of ticks so far. It's up to just September 1st, and it's 131 sample pools. But we are finding a significant number of American dog ticks in British Columbia, which, previously we had thought that the range had expanded into Alberta, but not as far as British Columbia. And so we really want to follow up this a little bit more, see if there are sort of focal pockets where we're seeing the American dog tick and its range is still limited, or if these are coming from across the province. And again, we're going to add significant more samples to this analysis, as well as samples from Manitoba, so we can really get a full understanding of where these ticks are being submitted from across the West and what the species distribution is. So stay tuned for that. I think there will be some interesting findings. And then this graph just shows life stage of tick submission, which at first you would look at it and it's not overly surprising. We know that our adult females are the predominant ticks that we find on companion animals. But what I find quite interesting is that there's a good proportion of male tick submissions. And there's even some nymph and some larvae. I'm incredibly impressed by the larvae because when I get them in the vial, I don't even think there's a tick in there. And so to be able to see that on your pet is pretty remarkable. But the males they're not engorged, and we don't think about them a lot on companion animals because we know that they generally don't feed for very long. They go onto a host to mate with the female, have a little blood meal, and then they leave. And so if you're going to see them, you're seeing them fairly soon once the tick comes on to the companion animal. And so what I think this is evidence of, in addition to, I don't have good proportions to share with you today, but probably about, I would estimate, a third of the female ticks we're seeing are also not engorged. And so what I think this is showing us is that people are actually looking for ticks on their pets, and they're finding them early on before the tick has the ability to engorge. And to me, this is really positive news. It means that the messaging that we've had about the importance of doing tick checks and making sure that when you come in from outside, you check your pet for ticks, is actually really making a difference. And so we're going to dig into this a little bit more to look at engorgement status of these ticks, but keep that messaging up. I think it's really important, and really extend that to humans as well. That tick checks for pets and humans are really important to get those ticks off early. The final results to share are just some preliminary pathogen testing. And so I only have that from Ixodes scapularis and some Ixodes pacificus, and not a lot. So these are samples from as of October 1st, so from April to October. So we're really missing the fall peak. And I know that some of our students have been busy in the lab over the last couple of months processing thousands of samples to get the results out. And so this is just very basic level data. In Ontario and Nova Scotia, there's enough data here that we have hundreds of samples that this is based on. And so we're a little bit more confident, as you can see from the confidence intervals, that this is a more reliable indication of infection prevalence. And certainly not really surprising. We know that these areas have well-established black-legged tick populations, and generally we see between 20 to 30% infection prevalence in those areas. Some of the other areas where we do have data, and you can see them a little bit higher, this isn't based on it's based on less than 100 ticks. You can see the confidence intervals are really wide, meaning we don't have a lot of precision in our estimates currently. And so this is just sort of showing you what we have so far, but we're going to have a lot more value in the data once we get a larger sample size. The other real value in the data is going to come when we look at sort of it on a regional level. So, for example, Ontario is a very large province. We see pockets where our prevalence is very different from one area to another. And so not only looking at this on the provincial level from a larger sample size, but also looking at the regional level to look at where our focal pockets of risk are will be really helpful. And that's a planned part of our analysis. And then finally, we have some results of Anaplasma. And again, we know that this is seen at a much lower infection prevalence than for Borrelia burgdorferi. And we know that there are some small areas where there are higher infection prevalence. So we're seeing a little bit in Eastern Ontario and some areas of Manitoba. But again, this is based on a very small sample size. As you can see, for Saskatchewan and Alberta, I think it's based on 1 tick. So we really can't make any predictions from that, which is why you see sort of a confidence interval all the way from 0 to almost 100. But again, as I said, with our Borrelia testing, once we can get this down to a regional level and have a bigger sample size, we'll be able to look at some different patterns that we're seeing and see if any risk is changing from previous data that we have. So just some take-home messages so far, and I think there's going to be a lot more as we're able to process a lot of the data and also look at a lot of our survey results that come in with the ticks, as we really haven't processed any of that data yet. We do know that there's significant differences in ticks and pathogen risks that exist over space and time. Canada is a big country with a lot of different ecological areas and climatic areas, and so looking at a regional level and having that sort of granularity to our data will be really important. We can already see some of those patterns emerge, but we're going to build on that and really hone down to the regional level in this study. Other message, cats get ticks too, just to sort of continue to debunk that myth that cats just take ticks off of them, because we're seeing that they're really not, and we should be thinking about our kitty cat companions. Based on our current evidence, we don't see any patterns that are suggestive of new tick species establishing in Canada. So when I mentioned the Lone Star tick and the Asian longhorn tick, none of our data supports establishment at this point. But range expansion does continue, particularly for our dermacenter species and our black-legged tick species, and we need to continue to monitor this. And the other sort of, I think, positive take-home message is that owners may be doing tick checks. They seem to be finding them before engorgement is happening. And that's a really positive message to keep encouraging owners to be doing these tick checks. And the next steps for us are just to continue to collect our data, to analyze to a much more in-depth level, looking at spatial and temporal patterns and those patterns of infestation. And then finally, which is probably the most exciting aspect of this project, is being able to communicate these results. And we've gotten funding from several industry groups to take this to take these results and produce regionally specific infographics for distribution to veterinary clinics across Canada. So please stay tuned for that, because I think that's going to be a really great way to get this research out of the lab and into the hands of veterinarians and pet owners who really need this information the most. And finally, I know I'm running close on time. I just wanted to sort of tie this all together. Many of you may be familiar with the One Health approach. It's sort of a buzzword a little bit in our profession, but it does have really strong value, and thinking about ticks and tick-borne disease in the context of One Health is really important. And so for those of you who aren't familiar, One Health is defined as a collaborative effort, collaborative being the key word there, of many disciplines working locally, nationally, and globally to attain optimum health of humans, animals, and the environment. And so we're looking at issues that have inherent overlap between environmental factors, human health factors, and animal health factors. And really at that intersection, we get the need to apply a One Health approach. If we think about ticks and tick-borne diseases, and we just think about them in sort of a traditional manner, we'd probably be thinking about transmission cycles. And so we know that there's transmission between wildlife reservoirs and tick species, and then ticks can spread those to humans and our companion animals. But there's a whole other host of factors and interacting factors that are going on. We know that the environment is incredibly important to supporting tick and wildlife populations, and that changes in our environment are leading to changes in the tick risks that we see. Even when there are wildlife species that aren't reservoirs for the bacteria, they play really important components in tick ecology, particularly white-tailed deer and the black-legged tick. And then we have interacting factors between all of these, including human and animal behavioral factors that influence risk, tick acquisition, tick bites, and use of prevention methods. And so in this context, using a One Health approach and bringing together different disciplines is really important if we want to be able to have a comprehensive understanding of the risks and design preventative and control measures that are holistic and look across the spectrum of the situation we're dealing with. And so with that, I'd just like to leave a few messages of where veterinarians can be involved, particularly with ticks and tick-borne disease. And I know many of you already are, based on the response that we've gotten from our Canadian Pet Tick Survey, which was overwhelming and extremely positive. We know that vets are thinking about this on a daily basis. I've included a few other surveillance initiatives that are going on. So depending on what province you're in, etick.ca is a great resource that helps with tick identification and provides maps of where ticks are being found. Myself and Drs. Weiss and Evanson run the Pet Tick Tracker on petsandticks.com, which anyone can submit to across Canada, and we produce maps and share the data that we're getting, and it's really a citizen science-based tool. And we're also working with the Canadian Lyme Disease Research Network, who's put together a national sentinel surveillance network, and we want to build in national veterinary surveillance into that network. And so that project is in its infancy, but stay tuned because that's really going to need collaborative effort from veterinarians across Canada. And then finally, education is a really key place where veterinarians can have a role in One Health. We know that ticks are shared risk between companion animals and humans. We visit similar habitats, particularly if we're going out and experiencing the great outdoors with our pets. And a lot of preventative methods are across the board. Of course, humans don't have a pill that they can potentially take every month, but still the importance of doing tick checks is across the board for animals and humans. Knowing where ticks are, educating yourself on distribution, and also avoiding tick habitat when possible. And those are all very similar preventative practices that are important for domestic animals and for humans alike. So having those conversations in practice about those shared risks can be really important. That's it for me, but I'd be happy to take any questions as we wrap up today.
Thank you, Dr. Clow. On behalf of the Canadian Veterinary Medical Association and Merck Animal Health, we want to thank you all for taking the time to join us today, and thank Dr. Lindsay and Dr. Clow for sharing their expertise with us as we move into National Tick Awareness Month 2020. Throughout the presentation, questions were submitted, and answers to the top 5 questions submitted will be shared back to the attendees in the coming weeks. Thank you again for attending today's webinar.